Initiation of DNA replication. So we know that bacterial DNA is circular DNA. So suppose this is the representation of bacterial DNA. So suppose this is one of the bacterial DNA. So in this bacterial DNA, we have to initiate the process of DNA replication. So what happens that in the circular structure of bacterial DNA, there is one point at which DNA replication can be started, and that point is called origin of replication. That is simply called ORI, O-R-I, ORI. So there is one major point is that in bacterial DNA, there is only one origin of replication, but in eukaryotic DNA there are many origin of replication. So one of the major point that you have to remember that bacterial DNA has got only one ORI. It has got only one ORI, only one origin of replication. So suppose this is ORI site. Suppose this is the ORI. This is ORI site of bacterial DNA. So for the initiation of DNA replication process, this ORI is recognized by a special type of protein that is called DNA protein. DNA protein recognizes this ORI. Origin of replication is recognized by DNA protein. So once DNA protein recognizes ORI, we know that this ORI is rich in 80 base pair. It is rich in 80 base pair. It is rich in 80 base pair, that is several AMP and thymidine nucleotides are present at the site of ORI. So once DNA protein binds at ORI, it will cause the breakdown of the hydrogen bond between A and T. So this DNA protein has a tendency to cause the lysis of hydrogen bond present between two strands. So this DNA protein causes the lysis of hydrogen bond between these A and T. So once hydrogen bonds between A and T are lysed or broken down by the help of DNA protein, this portion will slightly expand, and that is called replication bubble. So after the breakdown of the hydrogen bonds between this A and T, this part slightly expands and it will form another type of a structure that is called replication bubble. So we can see here after the breakdown of hydrogen bonds, this part slightly expands, and this expanded structure will be like this. So we are not going to draw the whole structure in the further figures. So we will just simply draw a segment of this figure. Suppose we can take up to this part, we can take one of the segments of this DNA, and by the help of this segment we can simply explain the process of DNA replication. So suppose after the breakdown of hydrogen bond, this part expands like this. So this is replication bubble. This is called replication bubble. So since it is slightly expanded part, so it is bubble-like structure. So this bubble still contains DNA protein which has caused the breakdown of hydrogen bond between A and T. So once DNA A protein forms bubble, now helicase enzyme comes and bind at the both corner of DNA replication bubble. So these are the helicase enzymes. So once helicase enzyme binds at the both corners of DNA, this we know that helicase is simply responsible for unwinding of DNA structure. So after the formation of replication bubble at origin of replication, helicase enzyme binds at both corners, and helicase enzyme tends to unwind the both strands of DNA. So due to the action of helicase enzyme, what happens that double-stranded DNA get converted to single-stranded form. So we know that when single strands are formed in larger proportion, then they may undergo re-annealing process. We know that the meaning of re-annealing is rewinding of DNA strands simply. So just to prevent re-annealing process, just to prevent the process of re-annealing, as soon as single strands are formed, these single strands are bound with a special type of protein. Those proteins are called single strand binding protein. Simply they are called SSBP. SSBP. So these are SSBP, that is single strand binding proteins. So the role of single strand binding protein is that single strand binding protein makes the single strands more rigid. So once single strand binding protein binds at the single strands of DNA, that makes single strands more rigid, and that prevents from the re-annealing of single-stranded DNA. So now, then what happens that after the unwinding of all these structures, in the next step, this parental strand will form one new strand along with it, and this another parental strand will form another new strand, DNA strand along with it, so that finally two daughter DNA molecules get separated or get formed. So in further point, we will be discussing only the half portion of this figure, because we know that when there is formation of replication bubble and when DNA helicase makes more and more single strands of DNA, new DNA strands will be formed against this parental DNA strand, and another new strand will be formed against this parental DNA strand. So the event that takes place inside this side, like in the left side, the event that takes place to the left hand side will be same as that in the right hand side. So for the ease of explanation, we are going to simply take half part of this process or this figure, and we will be explaining the elongation process by the help of the half figure.